This presentation is autosomal linkage for A-level, specifically the AQA specification, but should apply just as equally to any of the other exam boards. Some nice easy questions just to get you up and running. Take a moment to read through the slide, and once you've finished and you've got all your answers written down, unpause the video and we can progress on and see how you perform. So question 1, how many chromosomes does a human have? Well, the human has 46 chromosomes. How many pairs of chromosomes? Well, 46 is 23 pairs. How many homologous pairs of chromosomes does a human have? Now remember, homologous pairs refers to pairs of chromosomes that have exactly the same genetic material on them. So there's 22 of those. We don't count set 23. The reason we don't count set 23 is because set 23 is a sex chromosome, hence humans have two sex chromosomes, XX or XY. And how many autosomal chromosomes do humans have? Well, autosomal simply refers to any chromosomes except the sex chromosomes. So again, 23 pairs of 44 separate chromosomes. So previous presentation, we went through dihybrid inheritance. Make sure you go back over it if you need to. In a normal dihybrid inheritance, we have two heterozygous parents. You should get a 9:3:3:1 ratio. Again, if any of that sentence didn't make sense, go back over the dihybrid presentation before you go any further. We've also got what's called the law of independent assortment. And in the law of independent assortment, what it means is that any of the chromosomes with any of the alleles on them are equally likely to be passed on into a gamete. So just because we've had one particular allele and one particular chromosome, it doesn't have any influence over any of the other chromosomes, and hence we can randomly allocate all of the alleles when producing our gametes. Sometimes, however, what we end up with is we end up with genes. So again, we're looking at two different polypeptides, but they're actually found on the same chromosome. And when they're found on the same chromosome, it means that whatever allele you get for one of those genes is always matched up to the same allele for the other gene. Let's have a little bit more detailed look at what we mean by linked and unlinked. On the left side of the screen, you can see that there are two separate pairs of chromosomes, one in blue and one in yellow. And on each of these chromosomes, we have a gene, gene A or gene B. If we look at gene A, look at the uppercase A, if that is passed on to a gamete, there is absolutely no preference in terms of which of the two letter B's is also passed on to the gamete. And that's what I mean by law of independent assortment. Each of those alleles for each gene is allocated completely independent of each other. If you look on the right-hand side with the red chromosomes, and we've got genes D and E, genes D and E are linked. What that means is that whatever allele for D and whatever allele for E that appear on the same chromosome, they will always be passed on through generations. Now, when I say always, it's slightly more complicated than that when we bring in a concept called crossing over. And you'll see that actually we can have during meiosis some variation which jumbles up the letters. But as a general rule, that chromosome will only pass on the uppercase D and lowercase e, or will pass on the uppercase D and the lowercase e, and that will just carry through every single generation unless crossing over occurs. So in autosomal linkage questions, we are looking for any chromosomes except the sex chromosomes. We are looking for two genes occurring on the same chromosome, and we're looking for what at first might appear like a dihybrid inheritance, but we do not get that 9:3:3:1 ratio if both the parents are heterozygous. Remember, 9:3:3:1 only occurs if both parents are heterozygous. You can go through and do a dihybrid Punnett square and have a look at expected outcomes and match them across that way and see if there's any variation. However, generally in an A-level exam, they wouldn't ask you to do anything that complicated. So if they are going to get you to compare against ratios, it should be a 9:3:3:1, but it's not for definite. They may try and make a really awkward question for you. If the question is autosomal linkage, you will see a large variation away from your expected ratios. And we'll have a look at some numbers later on, so make that a little more clear for you. In this particular example, we do have autosomal linkage, but we are not going to have any what we call recombinants. And recombinants are what occurs when we bring in the concept of crossing over. So just to get us started, we're going to ignore that concept for a moment and just assume that the alleles are on the same chromosome, are connected together forever and ever and ever. The question states that A and gene B control two different physical characteristics. It doesn't give us what those characteristics are, doesn't really matter to the question. Allele uppercase A is dominant over allele lowercase a, and the same with B, which is the normal case. If it's upper and lowercase, it does mean dominant and recessive. If you're looking at codominance or multiple genes, then you would have these superscript letters. When these type of questions come up on the exam, I personally find it quite helpful to put a little kind of visual representation of the chromosomes somewhere around the page, just by drawing straight lines to represent the chromosomes and then putting the relevant alleles and genes onto the relevant chromosomes. It's up to you whatever way you want to do it. It's just the way it works for my brain. You may find a better system. Parent 1 has the two lowercase and the two uppercase alleles. But notice that the two lowercase a and b are linked on one chromosome and the uppercase a and b are linked on the separate chromosome. And that's the same with parent 2. So you've always got the dominant A and dominant B linked together, and you've always got the recessive A and the recessive B linked together. And the question is simply asking us to show what would the genotypes be of the offspring. In this example, I've used blue and pink and drawn particular shapes and put letters in it, and it's a lot more complicated just purely for visual purposes. When you're in an exam, a simple straight line to represent the chromosome and the letters written in beside. Again, you can label male, female, parent 1, parent 2 is more than enough to help you keep track of things. All I've done here is just make it a little more colorful just so you can follow it. In terms of gametes, because the two lowercase letters A and B are on the same chromosome, they are linked together. Therefore, you will always have them in one gamete if we ignore crossing over. And the same with the uppercase letters, they'll always go into the other gamete. This will only happen because we're ignoring crossing over. We're only getting two gametes. And exactly the same for the other parent, you get exactly the same two gametes. So now that we know the gametes produced by each of the parents, we can stick them into a simple Punnett square. And from there, we can work out all the possible combinations. Once we've worked out the possible combinations, the question only asks the genotypes, so you can stop there, but I'm just going to show you how you would carry on if it asked for phenotypes. Because it doesn't specifically say what the phenotype of A and B would look like, we can simply put it down in terms of a simple dominant, 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 recessive, recessive, dominant, or recessive, recessive. The four outcomes that you would get in your normal 9:3:1 ratio. Unlike the 9:3:1, however, what we end up with is a case of 3 where they're dominant phenotypes for both characteristics, both genes, and one case where they are recessive for both genes. You can display that as ratios, or of course you can display that as a percentage. Again, it would depend on what the question would ask. But you have to notice there is, unlike a normal dihybrid inheritance, we're only seeing two phenotypes. In a normal dihybrid inheritance where you had two heterozygous parents for both conditions, you would get your 9:3:1. That doesn't happen here. I would even go so far as to caution you about memorizing as a 3:1 ratio, because depending on how the question is worded and depending on which alleles for A and B are linked together on which chromosomes, you can end up with other variations on that. So you really do need to carefully work through the question to make sure you get to the correct endpoint. Now let's have a look at what happens when we bring in the concept of crossing over. When you get crossing over, you get what are called recombinants. And recombinants are where the normal combinations of the alleles and genes on the chromosome are fixed, but if they swap over and you produce gametes that you wouldn't normally find, that's a result of a recombinant and only occurs as a result of crossing over in meiosis. This is a good point that if you are starting to think, ooh, meiosis, I can't quite remember what that is, go back over your notes, go back through your presentations and stuff, and make sure you get yourself back up to speed on the concept of meiosis. We're not going to go into it in any great detail here, but obviously it is an important topic and is quite likely to come up on any exam in any particular year. But crossing over is a particular phenomenon that happens during the process of meiosis. And what happens is when we have the homologous pairs of chromosomes line up, sometimes by pure random chance, you have an overlap or exchange of genetic material between some of the chromosomes. Now that actual overlapping is called a chiasma. And when you get a chiasma, what happens is, what can happen rather, because sometimes it happens and you end up with no change at all, but what can happen is that allele combinations that you wouldn't normally find start to occur. So if you remember back to the previous example, we only had 2 gametes and 2 final phenotypes. This is how we can end up with the other sets of gametes. As you can see in the diagram at the bottom, in the 2 middle chromosomes, the uppercase A and the lowercase a have basically swapped over and exchanged, and we now have more variation caused by the recombination of the A gene with the rest of the chromosome. So a loci refers to a particular location anywhere along a chromosome, and genes are always found in the same loci on a chromosome. So for example, if you look at chromosome A, you can see uppercase, lowercase, it doesn't matter, it's always found on the same part of that particular chromosome. What we can do is we can use how often recombinants occurred when we start doing large breeding numbers with how close together certain recombinants are. Basically, if 2 genes are close together, if one crosses over to another chromosome, the chances are that the other gene will go with it. And you end up with the combination being kept together even though it's on a different chromosome. If, however, the genes are far apart, then it's much more likely that one gene will exchange and crossing over, and the other gene will remain and be unaffected by the same crossing over. And depending on how often that occurs, we can actually make an indication with how far away or how close 2 particular genes are on any one chromosome. So here's an example of the kind of question that you might get. It says, explain which of the following combinations of genes would you expect to produce with the least number of recombinants? Well, first off, you should remember the fact that the least number of recombinants will occur when the genes are closest together. And if you look there, you'll see that gene 2 and gene 3 are closest together. And if you say the answer is gene 2 and gene 3, then you're wrong, because it quite clearly says in the question, which of the following. So if you work through the list, you'd find that gene 3 and gene 4 are the ones that are going to produce the least number of recombinants out of the 4 options given. And if you finish there, you've lost marks because the question says explain. So you have to make sure that you clearly stated why 3 and 4 will give you the fewest number of recombinants. And it's simply by stating that the fewer number of recombinants will occur when they are closer together is all it will take to get that extra mark. But if it says explain, you must explain. It is possible to put a numerical value to the distance between the different genes relative to each other. If this was to come up in the exam, I would expect the exam board to give you this equation or a variation of it if they wish to use. But in this particular case, all we have to do is take the total number of recombinants and then divide that by a total number of organisms. If you look at the last 2 bullet points, it gives you an example, and it says you have 1,125 organisms, 4 of one recombinant, 54 of another recombinant, and then we have to identify if gene 1 is A or D, and then obviously gene 2 is the other one of A and D. So when we put our numbers into our calculations, it would be 4 divided by 1125 times 100, which gives us a value of 0.4. And for the other recombinant, we get 54 divided by 1125 against the total population. And we multiply that by 100 to give us a value of 4.8. What that means is that A and B have a higher percentage recombinant value than B and D. Therefore, A and B are further apart than B and D. As a consequence, if A and B are further apart, B and D are closer together, well then gene 1 must be A and gene 2 must be D. It is possible that you can just go to the original question and not have to do the calculations, because what you should have got from the previous part of the video was that the lower the number of recombinants, the closer the genes are to each other. So it would indicate with only 4 recombinants that B and D should be very close together, whereas A and B should be further apart. But the question may come up on percentage recombinants, so it is worthwhile just being familiar with it. What we can do now is have a look at what impact the idea of recombinance and crossing over has in terms of populations, phenotypes, and genotypes. All I would like to do at this point is to read through the information on the slide, look at the data, and extract as much information out of it as you can. Literally dig through every little bit of detail you can, every bullet point you can. Once you think you've got as much information out of this as possible, then we'll continue on with the video and we'll have a look and see what the key points are. So at this point, pause the video, extract the information that you can, and then we'll see how successful you were. So the first thing that you should hopefully have noticed is that the F1 generation are heterozygous for both gene A and gene B, and we're breeding 2 heterozygous parents. It would be a standard dihybrid inheritance and therefore should have produced a 9:3:1 ratio. If you then look at the numbers of organisms produced, you can clearly see that it does not follow a 9:3:1 ratio. With extremely high numbers of 2 phenotypes and very low numbers of the other 2 phenotypes. That is the indicator in a question that you're talking about autosomal linkage and not standard dihybrid inheritance. Now that we know it's autosomal linkage, what you want to do is try and identify which of the alleles for A and D genes are allocated to which chromosomes, so what's paired off with what. If you go back to the original parents, or the grandparents from the F2, it says clearly that the uppercase A and the uppercase D are both found together on one parent, and the lowercase a and lowercase d are both found together on the other parent. Therefore, the gametes for those grandparents must have contained the uppercase A and D in one particular organism, and in the other parent, it would have had the lower cases A and D. Because we're talking about linkage, that would carry through to the F1 generation. As we know the combinations of alleles on each of the 2 chromosomes from the grandparents, the original parents, we know that the F1 generation will have the same paired alleles for each of the genes. And if you look in the question, the second bullet point in the question clearly states that all of the organisms have the same phenotype. Therefore, there have been no recombinants occurring. And for the purposes of an A-level question, that is the norm, because if you start having recombinants in the F1, it completely complicates the F2 generation. So if they're going to do that, they won't ask you about the F2. In this particular case, the F1 all have the same phenotype, therefore it's the F2 that they're going to focus their attention on. So as the F1 all have the same phenotype, we are going to assume they all have the same genotype as the original grandparents, which means the same alleles for genes A and gene D are linked together in exactly the same way, and they pass them on to the F2, which would mean that you would expect to find the uppercase A phenotype, which in this particular case is purple, and the uppercase D phenotype, which again in this particular case is long pollen grains would be very, very high in number because they are linked on the same chromosome. We would also expect to find red flowers and short pollen grains, represented by the lowercase a and the lowercase d, also to be in very high numbers. However, we also know that recombinants would occur, which would mix up the upper and lower cases for one of the genes. So either the A's would cross over because of a chiasma, or the D's would cross over because of a chiasma. It doesn't matter as long as one of them swaps without the other, because if they both swap over, then the A and D uppercase and the A and D lowercase stay together anyway. So they have to swap over without the other one. When that happens, we get our recombinants, but they should only be in very low numbers because crossing over is a rare event. And if you have a look there, you can see quite clearly that we have extremely high numbers of the purple and long, represented by the uppercase A and uppercase D, and the red and short, which is represented by the lowercase a and the lowercase d. We do have the other combinations of phenotypes, but they're in very low numbers, 19 and 27, and these are the recombinants that have only occurred because crossover occurred during the production of those particular gametes. Now, this question could lead on to quite nicely into a statistical test. Which statistical test would be needed and why? Take a pause, have a think, and see what you come up with your answer. And hopefully, because you're looking at observed, inspected, and categoric data, you should have said a chi-squared test. It's unlikely they will throw a chi-squared test in with something as complicated as this, but it's not an impossibility, so just be careful and watch out for it. All you're doing is comparing with your observed and your expected. So to finish off, there are a number of terminologies and concepts that have come up all the way through this presentation. And what I'd like you to do is take each one in turn, pause the video, come up with the fewest number of words possible needed to explain that concept. The reason I say fewest number of words is because the fewest number of words means you're using the key terminology effectively, and it also means that you understand what you're talking about. If you take 16 sentences to explain something, you're not clear on it, and certainly it will show up in an exam setting. So take each one separately, and we'll start with autosomal linkage. So that is the simple idea that a type of inheritance where genes are located on the same chromosomes. So they're linked. Categoric data, remember categoric data is tied to the concept of chi-squared, and that's simply where data can be separated into groups. The F1 generation refers to the first set of offspring that are produced. So normally you have kind of the original parents, an F1 and an F2 generation in a question. You can have grandparents, parents, and offspring, and we use the term F1 to refer to the offspring from the first successful breeding, and then F2 is the next one, and F3, and so forth. Recombinance refers to the allele combinations that can occur but are not present on the original parents. So they're not the ones that the parents had originally, they have appeared as a result of a particular process. And that particular process is the next one, which is crossing over. And crossing over is something that happens during meiosis, and it results in alleles being exchanged between chromosomes. Please don't say genes being exchanged. It's alleles for those genes that are exchanged. And finally, gene loci. Well, a loci is simply a particular location of a gene on a chromosome. And that brings us to the end of the presentation on autosomal linkage.